Let's take a look at our catalytic receptors. With our catalytic receptors, we have two different kinds. We have the kind that has its own receptor tyrosine kinase, or the own kinase activity as part of the receptor, or we have our cytokine receptors, which do not have their own tyrosine kinase activity as part of the receptor, but instead it has a cytosolic tyrosine kinase instead. So those are the ones over here, our cytosolic tyrosine kinases. Those are your cytokines. And they are cytokine receptors. Those are going to respond to things like interleukin 6 and interleukin 1 beta and tumor necrosis factor alpha. So each different type of cytokine, which is a small protein that's produced by some of our white blood cells like our macrophages, will have its own specific receptor that it can bind onto on different cells and stimulate a bunch of inflammatory cascades. And the way it's going to stimulate inflammatory cascades is once it binds onto its receptor, it's going to activate the cytosolic tyrosine kinase. That we had Sark was one. And the most common one is JAK, Janus kinase. JAKs will bind to STAT proteins, which are just they're adapter proteins basically. So JAK-STAT pathway is our Janus kinase is going to activate STATs. The STATs will dimerize together, so you have two STATs stuck together. And then those STATs will go to the nucleus and stimulate transcription. And what we're stimulating with transcription is we're taking a specific gene, and we're in most cases with cytokine receptors, it's an inflammatory gene, and we're going to make more inflammatory compounds. So then we have our other type of catalytic receptors, or the ones that have their own tyrosine kinase activity, and they're known as receptor tyrosine kinases. There are three different parts of the receptor tyrosine kinases. We have the part that's going to bind the ligand. It's usually your N-terminal. Since all proteins have an N-terminal and a C-terminal, you have the part that goes through the membrane, the transmembrane region, and then you have the C-terminal, which is inside the cytosol, which is your tyrosine kinase. With our catalytic receptors, especially our receptor tyrosine kinases, we are going to regulate them by using enzymes that are going to turn them on or turn them off. So our tyrosine kinase is going to phosphorylate tyrosines, and that's usually going to turn on a cascade. So that's your on signal, is we'll phosphorylate the tyrosines. And then we can dephosphorylate it using another enzyme that's called tyrosine phosphatase, which is kind of your off switch. So kinases phosphorylate and phosphatases remove those phosphates. Once we phosphorylate a protein, that phosphorylate that tyrosine, that's going to stimulate recruitment of adapter proteins. There's lots of different adapter proteins. SH2, I think, was one that I'd shown you before, but there's another one that's called GRB2. I don't care that you know that. I don't care that you know this entire pathway of what's happening here. One thing I should point out here is that RAS from our G protein coupled receptors, RAS superfamily, is also part of the signaling process too. So it's involved with a couple different signaling pathways. So it's going to recruit some proteins, and that's going to eventually activate PI3 kinase. PI3 kinase, in its job, a kinase is just going to phosphorylate. It's going to phosphorylate phosphatidylinositol. It's going to go from PIP2, 2 phosphates, to PIP3, the more active one. So another phosphate makes it more active. That's going to activate AKT, which is also known as protein kinase B. And protein kinase B's job is to phosphorylate. And it's going to phosphorylate a protein that's usually involved with apoptosis, and it's called BAD, is the protein. When we phosphorylate this BAD protein, it's actually going to turn it off. Remember, phosphorylation can either turn on a protein or turn off a protein. In this case, it's going to turn it off. And its job is usually to stimulate apoptosis. But when we phosphorylate this BAD, it turns it off and it inhibits apoptosis. So our receptor tyrosine kinases a lot of times are going to be involved with kind of prolonging the life of the cell. So this is going to be deranged when we see this in cancer cells. Cancer cells have this happening all the time without even having something bind onto that receptor tyrosine kinase. And what can bind onto that receptor tyrosine kinase? Some of the ligands would be things like growth factors. Or hormones can bind onto, peptide hormones can bind onto them as well. One other recruiting or adapter proteins is the STAT protein, which we saw with the JAK-STAT pathway, can be utilized with that also. So with this receptor tyrosine kinase, once it's activated, so it's going to form dimers. Once our ligand binds onto it, our growth factor, whatever it might be, they're going to pair up and they're going to help phosphorylate each other. Remember, that's like our married couple. They're going to phosphorylate each other on tyrosines, and then that's going to signal this entire pathway happening. The last part of this is our insulin receptor. Our insulin receptor is a type of receptor tyrosine kinase, which we've already looked at insulin signaling. But once our insulin, which acts as a growth factor, binds onto our insulin receptor, it's going to recruit our IRS1, which is going to be one of our adapter proteins. And we have IRS1, which is widely expressed, IRS2, that's widely expressed, IRS3, that's found in adipose, pancreas, and liver tissues, and then IRS4, that's located in thymus, brain, and kidney. So once that IRS1 is phosphorylated, when it's phosphorylated in type 2 diabetes, it can't dock correctly to the insulin receptor and it can't send a signal. Once insulin binds to our insulin receptor, binds to our phosphotyrosines, it can also stimulate that PI3 kinase pathway. It's going to activate the RAS and STAT and help stimulate budding of our GLUT4 as well. So we have a lot of overlap with a lot of these different pathways, and once you turn on one, it has a tendency to kind of amplify the signal. And overall, what we're going to end up doing with a lot of these is signal transcription, transcription, transcription. So eventually, once we transcribe that gene, we'll translate it into a protein. So we have more of a specific type of protein that's going to affect the cell.